Great. Right. Well, thank you. Thank you, Sonia, for inviting me. I'm going to talk today about focus specifically on a recent paper that we published with a number of co-authors around the world, and primarily worked on while I was at NIWA, addressing the effect that Salp blooms have on passive carbon exports in the Southern Ocean, specifically in the region east of New Zealand. To introduce myself, my name is Moira Decima, and I'm assistant professor at the Scripps Institution of Oceanography, and I'm also the curator of the pelagic invertebrate collection. So to start this talk, I just want to give you a bit of background on kind of three topics. One of them is carbon export and the biological carbon pump. So the diagram that you're looking at here represents all of the different biological processes that move carbon from the surface ocean to the deep, the mesopelagic and the bathypelagic. And in this diagram, we're including both passive components of the biological carbon pump and the active ones as well. But for my talk today, I'm going to focus primarily really only on the passive or gravitational sinking part of the BCP. And that includes generally all types of particles that sink from the epipelagic down deep. And that can be in the form of aggregates, such as we have with phytoplankton, and it can be in the form of fecal pellets coming from zooplankton. It can also be in the form of, say, carcasses, but that's also something that we didn't quantify very well in this particular study, but can be very important. The second background I need to give you is on salps. I'm sure most people on this call are somewhat familiar with them, but salps are gelatinous zooplankton. They have a complex life history that includes a solitary, also known as asexual, oozoid organism that, when it encounters good conditions, starts to produce clones that are blastozoids, and these are aggregate organisms, and they form these chains. And it's when they produce these organisms that they can have really high population growth rates. And in that process is where they produce what we normally refer to as a blip. Salps also have really high filtration rates. So we've kind of nicknamed them the ocean vacuum cleaners, because they can kind of mow everything in their sight. I did hear, I think Larry Maiden at one point referred to them as the ocean Roombas as well. And then they can produce rapidly sinking fecal pellets. So the ability of these pellets to sink, like if they're large or dense, really depends on the species. So larger species will produce pellets that sink more rapidly, but they can have, because of this, a significant impact on carbon export and also on food web flows. We've been known probably since around the 80s that salps can contribute a lot to export, but there's only been a handful of studies that quantify how much of that, how much they contribute to carbon export, and also not very many that compare one same location, compare the export when there's salps versus when there aren't any salps. And so there's a past study from the Indian Ocean that found a salp swarm to export about 30% of primary production, which is really high value. And when they compared that to studies from other times in the same location, they found that to be about 6 times higher than when salps weren't present. A recent paper by Steinberg et al. In 2023 in the subarctic Pacific found that salps contribute significantly to export, but it was they increased export flux only about 12 to 18%, and were responsible for exporting about 6% of primary production. Another number of papers in the Southern Ocean found that salps didn't contribute a whole lot really to export, only about 1 to 3%. And I'll tell you the results about this study in a moment. But you can see that even though we think they're important and they can be important, studies haven't shown that that's the case across the board. I want to tell you quickly about the bloom evolution. So typical conditions when there aren't salp blooms, there are oozoids that are drifting and surviving, and they need enhanced food conditions to be able to trigger this bloom. So when they encounter these conditions is when they start this production of these chains of blastozoids that grow rapidly. Every one of these blastozoids gives birth to one live embryo, and that's how the cycle gets completed. And the last topic of the background that I want to quickly touch on is the study region. So this is in the Southern Ocean, but it is specifically the subtropical convergence zone in the Southern Ocean. And in this region around New Zealand, this coincides with this bathymetric feature called the Chatham Rise. And hopefully you can see in the animation in the back, it's showing you sea surface temperature kind of changing over time. And so you can see that it's a dynamic area where waters from the north that are subtropical and are warmer and are limited in nitrate meet waters from the south that are subantarctic and colder, and they're limited in iron. And it's that mixing of water that can produce an area of enhanced localized production. Past studies in the region have also shown that generally microbial processes dominate, so microzooplankton tend to consume most of the primary production. And past studies looking at export down to 1,500 meters on moorings set in subtropical waters and subantarctic waters also found the area to be generally characterized by low export. You can kind of see this in this figure, the time series of 12 years, but hopefully you can also see that they are times of episodic fluxes. And while the paper doesn't really determine what is causing these episodic fluxes, we can imagine that potentially salps could be a cause for them. We also know from a few studies that salps can be important in the region. This is a figure from Henschke in 2013 that is showing that many locations in which salps have been collected from fisheries trawls in New Zealand. And we also know from anecdotal evidence from when we've gone out on cruises that we recover these nets full of salps, that these organisms are kind of persistent features of the region. And we also know, because there are a number of fish species that are that feed exclusively on gelatinous zooplankton, we know that salps are a recurrent feature over the Chatham Rise. So with that knowledge, we designed an expedition to go out and study all the different processes that we thought that salps would be affecting. We called it salp particle export and ocean production, or salp poop. We were primarily concerned with their role in carbon export. But we were addressing, for this paper, I'm going to talk about two main questions, and one is, how do salp blooms alter carbon flux? And so notice that I'm talking not just how much do they contribute, but how much do they change the amount of export flux that would have or would have not have been exported in that region had they not been there. And the other question we also wanted to answer was, how do they alter prey microbial assemblages? We've talked about how they can have really high grazing rates, and they're also generalist feeders, so they eat a very large range of particles. This is very different from traditional zooplankton consumers, and so we were interested to know how that might be changing protistan and phytoplankton communities. So during this cruise, our sampling was Lagrangian. So we would put out these drifters. Here I'm showing you we had two sets of drifters. One had a sediment trap to collect sinking particles, and the other one had bags with bottles in which we had our primary production and dilution experiments. And we followed these drifters around for a number of days, and we conducted a slew of other measurements in that marked water parcel. And so in that marked water parcel, we would do tows for zooplankton and salp abundance and grazing estimates. Okay, so this is this paper came out just the start of this year. So this is a close-up now of the area of the Chatham Rise and of the 5 locations that we sampled. So remember, I want you to remember that this is the direction of flow. So during the cruise, we sampled 5 different locations, and I'm going to call them cycles or Lagrangian cycles, experimental cycles. The first one I'm going to call Salp SASC, so that stands for Subantarctic Southland Current. The Southland Current is a current that's coming south to north on the east of New Zealand. And so Salp SASC is a bit of a modified subantarctic water mass that has primarily subantarctic characteristics, but it's also modified. And this location, we stayed there for 7 days. And so after 4.5 days, we pulled out the sediment traps and put in a new set of sediment traps so we could divide the cycle into an A and a B period. And that's basically because we wanted to stay there longer and we couldn't leave the sediment traps up for such a long period. So we kind of got 2 different cycles within kind of one location. The second place we sampled was a subantarctic location with salps, and we call that Salp SA. The third location we sampled was in subtropical waters, and that one did not have high abundances of salp, so I'll call it non-salp ST. The fourth was a salp cycle within subtropical waters, and the fifth one was a location in subantarctic waters without salps, or non-salp SA. So you can see here that we have salp locations and non-salp locations, and we also have them in subtropical waters and in subantarctic waters. And we really wanted to know if the effect that they were going to have on carbon flux or on plankton processes, if it was the same or different, if it depended on the water mass. And so we were trying to kind of get both of those. Okay, so first let me summarize a little bit what the underlying water masses look like. So because we're trying to compare salp locations to non-salp locations, we're using the non-salps as kind of an idea of a control. But it wasn't that easy to find really similar water masses with salps and without salps. And it worked better for our subtropical cycles than it worked for our subantarctic. So here I'm showing you size fractionated chlorophyll with depth, subantarctic waters to the left. And so those 3 cycles, they had some similarities and they had some differences. The maximum chlorophyll was up to 1 milligrams per meter cubed. The contribution of the smallest size classes was similar across the locations. But for instance, we can see that in Salp SASC, which is our nearshore salp location, we have the greatest contribution of cells that are greater than 20 microns. For Salp SA, we have more of a nanoflagellate important community. And in the non-salp subantarctic location, you can see that it's really dominated by picoplankton. For the subtropical waters, the 2 cycles were much more similar. The highest amount of chlorophyll was in the non-salp subtropical location. And both of these had significant amounts of biomass in the nanoflagellate range in 2 to 20 microns, and not huge contributions of the large greater than 20 micron cells. When we looked at the composition, we saw more diatoms in salp subantarctic waters, and you can see that both in the chemotaxonomic pigments and in the DNA-based community composition. And we found some evidence for more primnesiophytes in subtropical waters and a bit more in non-salp subantarctic waters. We also wanted to look at the phytoplankton physiology. So I mentioned that subantarctic waters are limited in iron, and that is what tends to keep these communities dominated by picoplankton. So I'm showing you first the FV of FM and the QA lifetime, which are measures of the stress that phytoplankton feel over iron. And hopefully you can see that the 3 cycles that that aggregate together are the 2 subtropical cycles and the Salp SASC, which is our nearshore salp location. Those 3 locations didn't have a whole lot of evidence for iron limitation or iron stress, whereas our 2 salp subantarctic locations did show evidence of iron stress. And then finally, I'm showing you phytoplankton growth as indicated by permit production and biomass accumulation. And the biomass accumulation is that net primary production minus the amount of phytoplankton that's consumed by microzooplankton grazing. And so you can imagine that as the amount of phytoplankton that would be left to sink, you know, if nothing else is consuming it or nothing else is happening. And so for that, the highest NPP was in the non-Salp ST location, followed by Salp SASC and Salp Subtropical. And also the highest biomass accumulation happened in non-Salp ST, followed by the nearshore Salp Subantarctic location. Okay, so let's look at the Salp bloom, and I'll be reminding you of the direction of flow because that's going to become important when we try and interpret our results. So the Salp bloom was primarily made up of Salpa thompsoni, but we did find other species such as Pigia confederata and Tethys vagina and Thalia democratica, just as an area, Elea maglanica and Salpa fusiformis. And those species really depended on if we were in subtropical, subantarctic waters, we had somewhat of different species composition. So for our Salp SASC, our nearshore subantarctic location, we have, I'm showing you now the abundance as a function of size. The green bars show the Ozoid stage and the brown bars show the Blastozoid stage. So you can see in the early part of our very first cycle, we had lots of small blastozoids, intermediate blastozoids, and we also had a noticeable part of the community that were oozoids, which later on in that same cycle, because we had such high production of really small blastozoids, compared to the fact that we didn't have that many oozoids, we can see a kind of shift in that in the composition of the life history stage. When we moved offshore, we saw communities that had more of these larger blastozoids mixed with smaller blastozoids and not high abundances of the oozoids of the solitary organisms. And then our last cycle, we had the largest blastozoids. You can see how that peak is shifting and getting larger. And the oozoids that we're finding in this cycle now are very small and often still had remnants of placenta. So these are the oozoids that are kind of being born to the next generation. When we looked at grazing estimated from gut pigments and ingestion, again, using the same color scheme, we find really high values in our first cycle in A and B. In the bottom there, I have summed both the day and the night fecal pellet production estimates, and they're around 80 and 75 milligrams per meter square of carbon per day. And those are the highest ones we got, kind of similar between those two parts of the cycle. And then as we moved offshore, we still had pretty significant amounts of carbon being ingested, but kind of about half the values that we were seeing in the first part of the bloom or in our earlier cycles. So then we measured carbon export flux using sediment traps. And so these sediment traps were placed at 4 different depths in the water column. So first I'm showing you the carbon flux measured in the non-Salp locations, in the subtropical and subantarctic locations. So you can see the values are less than 50 milligrams of carbon per meter square per day, and they're somewhat similar between subantarctic and subtropical waters, although they're lower in subtropical water, in subantarctic waters. Then if we move on to the Salp cycles, this is our first Salp cycle, Salp SASC, our nearshore location. You can see it has enhanced export. As we move to the next location, to Salp SA, further offshore, we have even more flux. And our last cycle, the subtropical water one, but also the one, again, the one that we sampled the last, had the highest amount of export of all the different cycles. We also looked into the material collected of the sediment trap and identified intact Salp fecal pellets. So this is going to be a conservative estimate of how much Salps contribute via fecal pellets, because a lot of the fecal pellets can break up or become aggregates. So the measurements here are showing only the ones that were really clearly Salp fecal pellets in the sediment traps. And those range between 20 to 50% of the total particulate organic carbon flux across the different cycles. And as you can see, usually around half of that POC flux were clearly identifiable Salp fecal pellets. these are the absolute values of the contribution, ranging from 20 to 60 milligrams of carbon per meter squared per day. And so we wanted to know, okay, how much does Salp increase carbon flux? So if we take the most conservative estimate and we're saying, okay, 50% is their contribution, that's the absolute minimum is that they're doubling the amount of flux, and that's based on these intact pellet estimates. But the reason we designed this with a Salp and a non-Salp kind of as a control was to be able to have a different estimate that's not quite so conservative, and it's taking into account all the pellets and all the other aggregation and the other impacts that their fecal pellets might be having. And so when we compare the flux from Salp locations to non-Salp locations, we get that the increasing carbon flux when we have Salps ranges between 2 to 8-fold. So on average, Salps are increasing carbon export flux by about 5-fold. Another thing we can look at is not just the amount of primary production that's getting exported, but also what we call the T100, which is how it's an estimate of how matter is sinking. So on this plot, the EZ ratio on the y-axis is the particular organic carbon flux normalized by net primary production. And the x-axis is this T100. And the T100 is the amount of flux relative to what made it below the photic zone that sinks 100 meters deeper. So for instance, the North Atlantic bloom experiment that has a value of 1, what that means is that 45% of net primary production that sank below the photic zone, all of that also sank 100 meters deeper. So it's just going kind of straight to the bottom. And then, so that is the most efficient, I guess, of our locations of the world in terms of export. These are other areas where these measurements have been done. And you can see, for instance, in the Southern Ocean, in the Kiwi experiments, there's a significant seasonal effect on that T100, where particles in January are sinking much deeper than in December. And off the western subarctic, you can also see a decrease in export flux that happens over the course of the summer. So significant decreases going from July to August. And then you can also see this seasonal effect on EZ ratio and Ocean Station Papa. These two locations, Station Aloha and ECPAC, you can see are areas of generally low export flux in the middle of the Pacific Ocean. And I put them there for reference as I show you now the values for our study. Okay, so for our study, this is where our non-Salp locations lie. So close to Station Aloha and ECPAC and the subarctic Pacific. This is our first Salp location, period B of the first one, and our second, and then our third. So you can see that as time has gone by, as the bloom has evolved, we are progressively enhancing that EEZ ratio. And so what we think we actually did was that we sampled the bloom during different times, such that at the onset of it, even though we had the most amount of pellets being produced, we weren't getting the highest values of export, right? And as time passed and as water's moving offshore, we're starting to see kind of export or particle production from earlier in the bloom being exported later down or being exported downstream. And so that's what this schematic is summarizing. You can see at the top, there are cartoons that represent what we measured in terms of the abundance and the size distribution of animals and what we infer that to represent in terms of the pellets. So you can see at the beginning, we have a lot of pellets being produced, 70 to 80 milligrams of carbon per meter squared per day. But and then the black pellets show the amount of carbon that's sinking as fecal pellets. So you can see that the production of pellets is estimated to be highest at the beginning, but the export is much higher later on. And not only is the absolute amount of export flux higher, but you can also see these black arrows, which are Salp fecal pellets. Those are also more significant later in the bloom. So we speculate that this, even though we're following water and measuring it there is this effect of particles being transported as waters move up. And then we also found evidence for this effect to be important at a region level. So this that I'm showing here, this is DNA data. This is Salp leads that we collected in the photic zone, that we collected on sinking particles and sediment traps, that we collected in the photic and mesopelagic zone, and also we collected from the seafloor. And the one thing I want to draw your attention to is that in our non-Salp subantarctic location, we did find lots of Salp DNA reads in the water column, but also in the deeper water and also on the seafloor. And this is consistent with our results from our thorium approach to measuring carbon export. So this approach relies on chemical equilibrium, where you use thorium to estimate the amount of carbon export. And the main thing I just want to tell you about here is that the waters in the non-Salp subantarctic location were very depleted in thorium. And so what that implied was that there had been a high flux event that had happened prior to when we were there. And based on this DNA evidence, we think that was probably, maybe that was water that had once experienced a Salp bloom, maybe a few weeks prior. Okay, I'm getting to them, but I'm going to very quickly tell you how Salps alter play microbial assemblages. And so for one, I told you we measured primary production and the different grazing estimates. And so these bars are showing you those different rates. The gray bar is showing you primary production. The second white bar is showing you microzooplankton grazing. The third white bar is showing you mesozooplankton grazing. The black bar is showing you Salp grazing. And then the red is the calculated net rate of change of phytoplankton. And the top panels are showing you the Salp ones, and the lower ones are showing you the non-Salp locations. So you can see that when we had Salps, there was always a negative rate of change in the phytoplankton community. And in our location without Salps, in the subtropical location where we had a lot of primary production, you can see that there's a positive net rate of change when Salps are not there. And in the subantarctic location, it was small and negative, and kind of expected based on previous studies, where microzooplankton are the ones that tend to really graze the phytoplankton down. So we're finding that when Salps are there, they can really shift that fate of the primary production. And then the other thing we did is using DNA metabarcoding on 18S to look at persistent communities and 16S to look at bacteria and archaea. We tested to see if Salps were significantly associated with the different communities. And so we did find that the Salp, non-Salp category could explain some of the variance we observed, and this was significant. The primary explanatory variable was water mass, not too surprisingly, but Salps were also important. And so we don't know if what this means is that certain microbial communities lead to Salp blooms, or if Salp blooms modify the communities afterwards. We can't really tell that causality, but we can say that that association was significant. And so to summarize, how do Salps alter carbon export? They enhance it by a lot, 2 to 8-fold, on average about 5-fold higher, but definitely twice as high if you take our most conservative estimate of just the fecal pellets and the sediment traps. They export up to 45% of net primary production and lead to BCP efficiencies of 27%, which we saw on that graph. Those are amongst the highest ones that we measure. But we did find that all the fecal pellets are not rapidly sinking to death. And so we did find that kind of decoupling between when we estimated fecal production to be high and when it was actually being exported, that makes sense in terms of bloom evolution within an advected system. We also find this evidence for a regional enhancement of export flux, and using this approach of comparing a location with Salps versus without Salps, especially in the subtropical area, we're able to say Salps really increase export flux by 3 to 6-fold in this location. And we can disentangle that from the effect of high expert flux just because, say, we have high net primary frequency. And then for the microbial communities, we find that when salps are there, they can drive down the phytoplankton standing stocks, which if they're not there, the phytoplankton might be accumulating. And then we also found these significant associations that are consistent with these salp or non-salp classifications. And I didn't talk about this right here, but this is results from another paper where we find that the salps are having the highest impact on prey that are in the 3 to 30 micron range. And so they're likely competitors of microzooplankton in addition to crustacean zooplankton. And with that, I'd like to acknowledge collaborators and co-authors, Andres Gutierrez and Scott Nutter and Matt Pinkerton and Mike Stuckel and Carl Safi. I remember all of these. Really, it's all of my collaborators and co-authors at NUA that was a really great study. And yeah, and I'll be happy to take your questions.
Thank you so much. My God, what an effort. That's amazing. Even just finding those locations must have been, yeah, a lot.
It's stressful.
Yeah, I mean, yeah, you know, you have a certain time on the voyage and you're like trying to find the right spot. Alexis, I think you have a question.
Yeah, amazing study, Moira. I've like read it a few times and it's amazing, and there's so much information there, and I was happy to see it, like you presenting it in this way makes me feel like I learned even more. But I was curious, I might have missed something. So you mentioned that the larger individuals ingest larger and faster sinking fecal pellets, right?
Mm-hmm.
So how come at the Salp sub-Antarctic location that, I think, if I heard you right, was dominated by smaller blastozoids and oozoids? Or maybe there was larger, but how come it had the higher sinking pellet, the sinking in pellet carbon ratio?
So, okay, so for one, I'm inferring the pellets from the size distribution of the salps, and we do know that they produce larger pellets. I don't have an answer. I mean, so yeah, I don't have a concise answer for you in terms of like why we measured that. It's likely, I'm making this analogy of the blooms moving with the advection, but that doesn't mean that that last salp location that we sampled was the endpoint of that exact first one. Yeah, so I'm kind of comparing it within that context, but it would be great if you had followed the exact same one, then we'd be able to like kind of know that for sure.
Yeah, I just think it's so interesting because it's like, oh, maybe the smaller blastozoids and oozoids are contributing just as much as the larger sizes.
Yeah, well, and I think that aggregation, like if you give it time, can also kind of play a role there. Because we weren't measuring. I'd have to ask Mike if he has results on this. I haven't seen results of like the size composition of those pellets measured in the sediment trap, because that would go kind of directly towards that question of, is it more pellets or is it larger pellets that are sinking, right? Because that's what you're asking.
Yeah, exactly. That's what I'm trying to get at.
Okay. Yeah, I should ask Mike if he has estimates of that. I would think so, but I haven't seen that data.
Okay, cool. Awesome. Thank you.
Okay.
I think I had a similar question to like, do you see that the pellets fall apart easily in the traps, or are there kind of like, because on the Solas voyage in 2020 we had some salps, salp bloom, and we had lots of like fluffy parts that could have been like degraded or slowly degrading salp pellets, but I'm not sure how well they conserve kind of over time. I guess your sediment traps were not out there for such a long time, but well, I don't know. Ours were not out there for a long time either. How stable are the sediment traps? Do you know?
Well, so the ones that were counted would be ones that were just really clearly square shaped. Yeah. But there are all of the ones, like either the ones that are more mucousy, like the the type 1, 2, 3 pellets described in Iverson et al. I mean, they do produce those. And then you can see, you know, their pellets are also like the string that is all put together, and that also starts to like unravel. So yeah, I mean, I think they're fast thinking, but I also think it's, you know, what's happening in the ocean is different than what we're seeing in the lab.
Yeah, sure.
And they are kind of like breaking up more. For sure.
Interesting.
I had I just want to comment to that, that I was chatting with Dave Karen, because Dave Karen and Larry Maiden in their early days of working with Salps and diving with him. And apparently Larry Maiden told him that he saw Salp poop in action. And so he's like, I'm gonna follow it. And he followed it, and the pellet never sank.
Wow. This could be the next tourist attraction, you know, instead of diving in dolphins, diving in Salps, you know, it's so much more fun.
I mean, I would pay for that.
Totally. Oh, that's nice. Does anybody else have another question? All right, doesn't look like it, but okay, well, in that case, thank you so much for your talk, and also Claude earlier for your talk. Yes, yeah, it's great to have these seminars going on. I learned so much about the papers, even though I read them before. It's just, yeah, it's great. Thank you so much for taking the time. And if you know, if you have any other kind of mesopelagic work related coming out soon, like, just feel free to contact me or Adrian Martin in the UK to, if you want to present. He's looking for a platform to present it. I'm always looking for speakers. Thanks everybody. I will stop recording now.